So imagine looking at a sprawling city from space. Like from far away through the clouds, it really just looks like one solid gray mass, or
Yeah.
it's totally monolithic.
Right, just a blur on the map.
Exactly. But as you zoom in closer, you start to see all these distinct features. You see an industrial zone pumping out smog, a massive highway network moving supplies, a central grid pulling electricity.
It's structured. It's not just a blob.
Yeah, and usually when we talk about a medical diagnosis, there's this expectation of binary precision,
Right.
like a broken bone on an X-ray. It's either broken or it's not. But then you look at cancer, and specifically brain cancer, and for the longest time, we really viewed tumors like that blurry city from space, just a solid lump of mutant cells.
Yeah, I mean, it was a necessary simplification for a long time. We always suspected tumors were more complex, but we simply didn't have the cartography tools.
Right.
We couldn't see the individual streets and the buildings. We were treating this incredibly complex ecosystem as if it were just a bag of rogue cells.
Well, today we are taking a deep dive into a study that finally gives us that map. We're looking at a pretty groundbreaking April 2026 paper from Nature Neuroscience.
It really is a landmark paper.
Yeah, the target of this research is glioblastoma, or GBM, which is the most prevalent and aggressive primary malignant brain tumor in adults. So the mission for us today is to explore this tumor not as a generic lump, but as a highly organized, complex cellular society.
A literal microscopic city.
Exactly. And the researchers mapped this microscopic city using this massive data pool, like 121 spatial and single-cell profiles from 100 different patients.
Okay, let's unpack this. How do you even begin to map a microscopic city? Because historically, didn't we just grind tissue up to study it?
We did, yeah, and that's actually the perfect place to start. Traditionally, if you wanted to know what genes were active in a tumor, you took the tissue sample and essentially blended it into a molecular smoothie.
A smoothie. Sounds appetizing. Right. You sequenced the RNA, which gave you a great inventory of what cells were present. But you completely lost the context of where those cells lived in relation to one another.
Oh, I see.
Like you knew a baker and a blacksmith lived in the town, but you didn't know if their shops were next door to each other.
Which, that matters a lot if the blacksmith keeps accidentally setting the bakery on fire.
Precisely the issue. So to solve this, the researchers utilized a revolutionary technology called spatial transcriptomics, specifically a platform called 10x Visium, alongside some very advanced in situ hybridization.
Okay, so how does that work? Visium spatial transcriptomics.
Let's look at the mechanics. Instead of a smoothie, you take a vanishingly thin slice of intact tumor tissue and you place it on a very special glass slide.
Just one thin slice, keeping it totally intact.
Exactly. And this slide is printed with thousands of microscopic spots, and each spot contains millions of unique DNA barcodes.
Okay, wait, so the slide itself is kind of like a giant grid of QR codes?
Think of it exactly like that. When the tissue slice rests on this grid, the RNA molecules from the cells drop down and bind to the barcodes directly beneath them.
Oh, wow.
So when you sequence that RNA later, the barcode comes along for the ride. You aren't just getting the genetic identity of the cell, you are getting its exact GPS coordinates within the brain.
That is wild. They essentially built Google Earth for a brain tumor.
They really did. And to get even more granular, they used in situ hybridization. That's where they send in fluorescent probes that physically light up specific RNA molecules right there in the intact tissue under a microscope.
So you can zoom all the way down to see individual cells glowing in their actual neighborhood.
Exactly. But when they finally zoomed in, the first major hurdle was simply figuring out where the city limits actually were.
Right, because where does the healthy brain end and the tumor begin?
That's the problem. Glioblastoma cells are notoriously good at infiltrating healthy tissue. They don't form a neat, clean boundary.
So how did they draw the borders? Because I imagine under a microscope, a cell is just a cell, right?
Well, they used advanced spatial algorithms, things like cell-to location and spatial. They used these to look for specific chromosomal errors or copy number variations.
Copy number variations, meaning they have the wrong number of chromosomes.
Yes. Healthy cells have a strict number of chromosomes, but cancer cells are chaotic. In glioblastoma, the classic hallmarks of this chaos are having an extra copy of chromosome 7 and missing a copy of chromosome 10.
Oh, interesting. So just a +7 -10 situation.
Right. By tracking those specific genetic typos across the spatial map, the algorithms could draw a definitive, data-driven border between the healthy outer tissue and the malignant core.
Okay, wait, let me challenge this for a second. We're calling this a city with neighborhoods, but how do we know these are actual organized communities?
What do you mean?
Like, couldn't these just be random clusters of mutant cells that happen to look similarly just because of where they randomly landed in the brain?
That's a really vital piece of skepticism. And that's exactly what the algorithms were testing for. The researchers weren't just looking for physical proximity. They were looking for statistical recurrence across 100 different patients.
Meaning they saw the exact same patterns over and over.
Yes. They found that these cells aren't just bumping into each other randomly. They organize into 4 distinct cellular communities, or CCs.
So dynamically interacting zones.
Exactly. Zones defined by consistent repeating patterns of gene expression. And the malignant cells themselves fall into 4 main flavors based on the healthy brain cells they're trying to mimic.
Let's list those out for the listener.
Sure. There are mesenchymal-like or MES-like cells acting like wound-healing tissue. There are astrocyte-like or AC-like cells mimicking the brain's support stars. Then oligodendrocyte progenitor-like or OPC-like cells. And neuroprogenitor-like or NPC-like cells.
Okay, so before we zoom in further, let's just pause and make sure we have the blueprint so far for you, the listener.
Good idea.
First, glioblastoma isn't a random mass. It's highly structured, organized into 4 distinct cellular communities, or CCs.
Right.
Second, researchers mapped this using spatial transcriptomics, which gives us the exact GPS coordinates of different cell types by tracking their missing or duplicated chromosomes, specifically chromosomes 7 and 10.
Spot on.
And third, the tumor cells are classified into those 4 main flavors, MES-like, AC-like, OPC-like, and NPC-like, based on their genetic programming.
Exactly.
So listener, here's a question for you to chew on. Think about a thriving city. If you were a tumor trying to survive and conquer a new territory, what kind of infrastructure or neighborhood would you need to build first?
That hypothetical is exactly what the researchers observed in the first 2 communities. They focus entirely on survival and infrastructure. And this brings us to a really fascinating divergence in the tumor strategy.
Let's get into it.
The paper details how the MES-like tumor cells, the ones mimicking wound-healing tissue, actually split into 2 completely different subpopulations based entirely on where they live.
Ah, the tale of the 2 twins. Let's look at the first twin, living in what the researchers labeled Cellular Community 1, or CC1. This is the hypoxic district.
Right, and hypoxic just means low oxygen.
Right. I imagine as a tumor grows super fast, it outstrips its own blood supply, creating these dead, suffocating zones right in the center.
That's exactly what happens. The environment inside a rapidly growing tumor is incredibly harsh. In CC1, the oxygen levels just plummet. A normal cell would undergo apoptosis or programmed cell death and clear out.
But the tumor adapts.
It does. Here, researchers found a specific subpopulation of these cells that we'll call MES-HIP. They have a very specific genetic barcode tailored for surviving in a swamp. They are CD44 positive, LIF positive, and IL-11 positive.
Wait, what are they actually doing to survive, though? Just like holding their breath?
No, they're sending out distress signals to hire mercenaries. The spatial mapping showed that these MES-HIP tumor cells are physically clustered right next to a specific type of immune cell called a tumor-associated macrophage.
Okay, macrophages. Those are usually the body's garbage collectors, right? The frontline immune defenders.
Exactly. They're supposed to eat things that don't belong. But the tumor cells secrete a specific chemical signal, a protein called VGFA.
Let's break down that mechanism because this is where the biology feels almost sinister. Chemical signaling usually works like a key in a lock, right?
That's exactly the right framework. The MES-HIP tumor cell holds the key, which is the VGFA molecule. It remises this key into the surrounding swamp.
Okay.
And the key floats over to the macrophage, which has specific receptors, NRP1 and NRP2, on its surface. Those are the locks. They also use another pair, MIF binding to CD74 and CD44.
And when those keys turn the locks, the macrophage just gets brainwashed. The tumor cell is literally hijacking the immune system, forcing the body's own defenders to act as its personal bodyguard in this low-oxygen wasteland.
It's incredible.
It feels like a hermit, a hypoxic hermit, just hiding in the suffocating swamp of CC1, frantically throwing out these VGFA keys to hire thugs to protect it from the rest of the immune system.
The hypoxic hermit is a brilliant way to conceptualize it. It's a purely defensive survivalist strategy. But if we travel over to cellular community 2, we find the other twin. And its strategy couldn't be more different.
Okay, so CC2 is the vascular rich district. This is the tumor's highway system.
Exactly. Here, we don't find the hermit, we find the ast variant of the cell. They have a different profile, CD44 positive but alive negative, and expressing genes like CO1A1 and CO1A2.
And since they are in the vascular district, I'm assuming they are sitting right next to the blood vessels.
Intimately wrapping around them. They surround the endothelial cells, the pericytes, and the vascular smooth muscle cells that make up the blood vessels.
Wow.
And because they aren't trying to survive in a low oxygen swamp, their method of communication, their keys and locks are entirely different. They aren't using VEGF-A to recruit immune bodyguards. Instead, they are expressing collagen formation genes.
Collagen, like the structural protein in our skin and joints.
The very same. These tumor cells secrete specific collagen proteins, like COL1A2 and COL5A2, that act as keys. They unlock receptors like ITGA1 and ITGB1 on the blood vessel cells.
So they're literally laying down a fibrous, collagen-rich extracellular matrix.
Right. They are actively reinforcing the physical plumbing of the tumor to ensure a steady supply of nutrients.
Ah, so this twin isn't a hermit at all. It's more of an astute architect. It lives in CC2, sitting right next to the highways, quietly snapping together collagen roads to keep the supply lines open.
Exactly.
That is a massive distinction. You have the hypoxic hermit in the swamp and the astute architect on the highway.
And the clinical implications of this distinction are profound. For decades, oncology has targeted quote-unquote tumor cells as a broad category, but the hermit and the architect are doing completely different jobs, using completely different tools.
Oh, I see where you're going with this.
Right. If you use a drug that blocks collagen formation, you might disrupt the architect, but the hermit in the swamp won't even notice.
That perfectly sets up our recap for this part of the map. First, these tumor cells are not a monolith. They have distinct subpopulations with entirely different jobs depending on their neighborhood. Second, CC1 is the hypoxic zone, where our hermit cells, the MES-HIP variant, recruit immune macrophage bodyguards by sending out VEGF-A signals.
Correct.
And third, CC2 is the vascular zone, where our architect cells, the MES-AST variant, interact with blood vessels using collagen pathways to build structural support.
Perfect summary.
So listener, here's your question. If you were designing a therapeutic drug to stop this SETI from growing, how might targeting the architect's collagen highways impact the tumor differently than blocking the hermit's bodyguard signals?
It really forces us to realize that monotherapy using a single drug is likely destined to fail against an organized city. You need a coordinated strike. But as we move to cellular community 3, the stakes get even higher.
Oh, yeah.
If CC1 is the swamp and CC2 is the highway, CC3 is the hacker collective.
I have to say, this part of the paper completely shifted how I view cancer. So in CC3, the mapping showed a dense cluster of tumor cells, specifically the ones mimicking oligodendrocyte and neural progenitors, the OPC-like and NPC-like cells.
Yes, gathered right around healthy brain neurons.
And the old way of thinking was that a tumor acts like a bulldozer. Right. It grows, it takes up space, and it just physically squishes the healthy brain tissue out of the way.
Yeah, that was a standard model.
But that's not what the spatial map revealed.
Not at all. The bulldozer theory is officially obsolete. These tumor cells are not just pushing neurons aside, they are physically plugging into them.
Mind-blowing.
And to prove this, the researchers didn't just rely on the spatial maps. They deployed a highly advanced, incredibly delicate laboratory technique called patch-seq.
Okay, we need to slow down and explain the mechanics of patch-seq, because when I read this, it sounded like actual science fiction. How do you measure the activity of a single cell?
It requires microscopic precision. In a traditional patch clamp experiment, a scientist uses a glass pipette. It's a tube so impossibly thin that its tip is measured in micrometers.
So basically invisible to the naked eye.
Right. Under a high-powered microscope, they carefully lower this pipette until it touches the outer membrane of a single living cell. They apply a tiny amount of suction to form a tight seal, and then they rupture just a microscopic patch of that membrane.
Wait, so the inside of the glass tube becomes continuous with the inside of the cell?
Exactly. The fluid inside the pipette is now touching the fluid inside the cell. Because the cell membrane is full of charged ions flowing back and forth, the scientists can now use an electrode inside the pipette to record the actual electrical currents flowing across that single cell's membrane.
Using techniques like V-clamp and I-clamp.
Right. Yes. They're literally wiretapping the cell's electrical chatter in real time.
But the technique in the paper is patch-seq. What does the seq add to the wiretap?
Seq stands for sequencing. After the scientist finishes recording the electrical spikes, they use that same microscopic glass pipette to literally vacuum up the internal liquid contents of that specific cell.
No way. They just suck the cell's guts right out.
Pretty much. They take that fluid and sequence its RNA. So for the very first time, you have the live electrical behavior and the exact genetic identity of the exact same cell simultaneously.
Which is how they caught the tumor in the act. When they wiretapped the tumor cells in CC3, what did they actually hear?
They recorded spontaneous excitatory postsynaptic currents, EPSCs. They recorded electricity flowing directly into the tumor cells.
Let me just underline how paradigm-shattering that is. EPSCs are the electrical jolts that healthy neurons use to talk to each other across synapses. A synapse is the junction between two nerves. Tumor cells are not nerves. They shouldn't be receiving synaptic electrical currents.
They absolutely shouldn't. But the RNA sequencing part of the patch sec revealed the mechanics of the crime. It was specifically the tumor cells mimicking oligodendrocyte progenitors, the OPC-like cells that were forming direct electrically active synapses with healthy neurons.
Neuroglioma synapses.
Exactly.
So a healthy neuron fires a thought or a motor command, and the tumor cell actually receives the electrical jolt. How is that physically possible? What are the keys and locks allowing a mutant cell to plug into the brain's grid?
The spatial transcriptomics data pinpointed the exact mechanisms. The tumor cells are expressing specific normal synaptic building blocks to trick the healthy neurons.
Like what?
Well, they use highly specific ligand receptor pairs, things like NRXN1 binding to NLGN3 or EFNB3 to EPHB3, and a major one is a signaling protein called GDF11.
Okay, so the tumor cell presents this GDF11 key.
Yes, and it perfectly fits into a receptor lock ACVR1B on the healthy neuron. By turning that lock, the tumor cell essentially convinces the neuron to build a synaptic bridge right into the tumor mass.
The tumor isn't a bulldozer, it's a parasite tapping into the city's power grid. It's hijacking the brain's own electrical system. And what is it using that electricity for?
Growth. If we connect this to the bigger picture, the neuronal electrical activity acts as a microenvironmental driver for the glioma's progression. The electricity fuels the tumor's cell cycle, drives its proliferation, and powers its invasion deeper into the brain.
Unbelievable.
It forces us to view the tumor not as an isolated mass, but as an integrated rogue part of the neural network itself.
Which is terrifying, but also incredibly illuminating. Let's recap this hacker network for you, the listener.
Let's do it.
First, CC3 is a region defined by synaptic signaling and rapid cell proliferation, operating like a hacker collective.
Yep.
Second, specific OPC-like tumor cells form direct, electrically active synapses with healthy neurons by tricking them with specific protein keys like GDF11.
Exactly.
And third, researchers proved this definitively using PatchSec, literally wiretapping the cell to record actual electrical currents, or EPSCs, while extracting its RNA to confirm it was cancer.
Perfect.
So think about this. Knowing that a tumor can physically wire itself into your brain's electrical network, does this change how you view cancer? From a passive growing lump to an active parasitic machine.
It really demands a complete reimagining of our therapeutic approach. And just to briefly complete our map, the researchers also identified CC4.
Right, the fourth community.
Yeah, it's a mixed residential region predominantly composed of astrocyte-like tumor cells, serving as sort of the general bulk filler of the tumor mass. But the most actionable clinical value lies in disrupting the infrastructure we mapped in communities 1, 2, and 3.
Right, so let's bring this all together. What does this all mean for you, the listener, or anyone facing a diagnosis like this? We've explored the hypoxic hermit hiding in the swamp, the astute architect building collagen highways, and the hacker tapping into the electrical wires.
Yeah, it's a whole new paradigm.
The ultimate takeaway here is that treating glioblastoma and likely other cancers soon can no longer be about just dumping chemotherapy into the body, hoping to poison the dividing cells.
Exactly. If you only attack cell division, you are just knocking down buildings. The city's infrastructure remains totally intact, and it will simply rebuild. You have to disrupt the coordination.
Makes sense.
But because we now have this incredibly detailed multiomic map, we have a precise list of vulnerabilities. By identifying these specific keys and locks, like the GDF11 signaling between neurons and the hacker cells, or the VDFA signals recruiting bodyguards in the hypoxic zone, scientists have an entirely new atlas of therapeutic targets.
It's the dawn of true spatial precision medicine.
It is. This raises an important question regarding how we approach all solid tumors moving forward.
You don't just bomb the city, you jam the locks, you cut the communication lines.
And this spatial framework won't stop with brain cancer. Consider how breast, lung, or pancreatic tumors might be organizing themselves into similar complex interacting neighborhoods. The tools we discussed today will undoubtedly be used to map and dismantle those cities in the coming years.
It's a lens of clarity we've just never had before. All right, to wrap up our deep dive into this microscopic city, I want to leave you with one final, slightly haunting thought to ponder on your own.
Oh, this should be good.
We talked about how the tumor cells in the hacker district use those synaptic keys to physically wire into the healthy brain, receiving electrical currents to fuel their growth.
Right.
But in healthy biology, synapses are almost always two-way streets of communication. If tumor cells are capable of forming functional electrically active connections with our healthy neurons, what kind of information, if any, is the tumor actually receiving from the brain?
Oh.
Is the tumor just mindlessly stealing raw electrical power? Or in some strange, uncharted biological way, is the city listening to the host?
A profound and, frankly, deeply unsettling biological question. The more we map, the more we realize how much we still have to learn.
Thanks for taking this deep dive with us. Keep questioning the world around you.